Welcome back to G Fun Facts Online. Today, we are abandoning the macroscopic view of the world to dive into a microscopic drama that plays out every single second on every single leaf of every single plant on Earth. If you were to stand in a cornfield in mid-July, looking out over that green ocean swaying in the wind, it would look peaceful, static. But that is an illusion. Beneath that placid surface, there is a frantic, mechanical rhythm keeping our biosphere alive. We are talking about stomata, the tiny kidney bean-shaped mouths on the undersides of leaves. These are the gatekeepers of our planet. They are the lungs of the terrestrial world, the only way carbon dioxide gets into the food web to become sugar, and the primary way oxygen gets out to fuel our lives. But for centuries, these pores have held a secret. We knew they opened, and we knew they closed. But the precise choreography of that dance, how they react in the split second a cloud passes over the sun, or a dry wind hits the crop, has been a mystery. Until now. Today we are discussing a massive technological breakthrough from the University of Illinois Urbana-Champaign called Stomata In-Sight. This isn't just a better microscope, it is a time machine and a teleportation device for plant biology, allowing us to watch plants breathe in real time. But before we unpack the technology, we have to understand the high-stakes gamble every plant makes every day. It is called the transpiration compromise. Plants are stuck in a paradox. They are stationary organisms living in a hostile environment. To grow, they need carbon dioxide from the air. But the inside of a leaf is wet, and the air outside is usually dry. Opening the door to let carbon in inevitably lets water out. This is the fundamental trade-off of plant life. If a plant keeps its stomata open too long, it starves of water and wilts. If it keeps them closed to save water, it starves of carbon and stops growing. Evolution solved this with the stoma. It is a pore bordered by two guard cells. These cells are hydraulic marvels. When the plant wants to open the pore, it pumps potassium ions into the guard cells. Water follows the ions, swelling the cells like balloons. Because the inner walls are stiffer than the outer walls, they bow outward as they inflate, creating an opening. To close, they dump the ions, the water leaves, and the pore snaps shut. It sounds simple, but this system is incredibly dynamic. Stomata respond to light, darkness, CO2 levels, humidity, and drought stress. The problem is, our understanding of this dynamic has been fragmented. We have been suffering from a sort of biological Heisenberg uncertainty principle. In the past, if we wanted to see the stomata, we had to alter them. Early botanists would paint leaves with clear nail polish to make a mold, peeling it off to count the pores. That gives you a static snapshot, not a movie. Later, we used microscopes, but to look at a leaf under high power, you often have to cut it, mount it on a slide, and blast it with bright light. That stresses the plant out. The stomata close or behave erratically because the plant is dying on the slide. You cannot simulate a hot Kansas afternoon on a microscope slide. On the other hand, we had machines called gas exchange analyzers. These are clamps that attach to a leaf and measure the air going in and out. They are great for data. They tell you photosynthesis is happening, or water is being lost. But they are black boxes. A sensor might tell you that water loss dropped by 20%, but it cannot tell you why. Did all the stomata close a little bit? Did half of them close completely while the others stayed open? We were blind to the mechanism. We had to choose between seeing the structure without the function, or measuring the function without the structure. The Stomata In-Sight system was born from the refusal to choose. The team at the University of Illinois created a technological trinity. It is a bionic setup that merges optics, sensing, and environmental simulation. The first pillar is laser scanning confocal microscopy. Unlike a standard microscope that produces a blurry image of a thick object, a confocal microscope uses a focused laser beam and a pinhole aperture. It optically slices the leaf, building a crisp, three-dimensional image of the living guard cells without cutting the leaf. It is non-invasive. They can look into the leaf tissue of a living, intact plant. By using specific wavelengths, they make the guard cells glow against a dark background, providing high-contrast video of their swelling and shrinking. The second pillar is leaf gas exchange sensing. While the laser watches the cells, the leaf is enclosed in a specialized chamber. This is a feat of engineering. Standard chambers are bulky and opaque. This team engineered a chamber with an optical window transparent enough for a laser to shoot through, yet sealed tightly enough to measure gas flux with parts per million accuracy. This means that at the exact second the laser records a guard cell shrinking by 2 microns, the sensor records the corresponding drop in water transpiration. The third pillar is environmental control. They can play God with the weather inside that tiny chamber. They can control light intensity, temperature, humidity, and CO2. They can program the system to mimic a cloud passing over the sun, or a sudden heat wave. This allows for dynamic benchmarking. They can test the reflexes of the plant. How fast can a corn plant close its stomata when the air suddenly dries out? Does it take 5 minutes? Or 20? In a rapidly warming world, that reaction speed, the kinetic response, is the difference between life and death. So what happened when they turned this machine on? They looked at maize, or corn, one of the most important crops for global food security. The system revealed that stomatal movement is not a simple on-off switch. It is a complex hydraulic oscillation. They saw stomatal patches. These are clusters of pores that open and close in unison, sometimes behaving completely differently than their neighbors just millimeters away. This explained the erratic data we used to see in the old sensors. But the biggest finding was the lag time. When light hits a leaf, photosynthesis wants to start immediately. It demands CO2. But the mechanical process of inflating those guard cells with water takes time. The laser imaging showed that in some corn varieties, the stomata were sluggish. They took several minutes to fully open. During that lag, the plant is missing out on growth. Conversely, when a cloud passes or the sun goes down, some stomata are slow to close. They stay open, leaking precious water into the dark night for no gain. This sluggishness is a major inefficiency. This is where the agricultural revolution begins. We are currently facing a yield plateau in major crops like wheat, rice, and corn. We have maximized the size of the grain. The next gains must come from making the plant a better machine. Using Stomata In-Sight, breeders can now screen for plants that have snappier reflexes. We need plants that open wide instantly when water is plentiful, but snap shut the moment stress hits. This is critical for water use efficiency. Agriculture consumes 70% of the world's fresh water. A plant that is slow to close during a 10-minute cloudy period is wasting water. Over a growing season, those minutes add up to gallons. A smart plant preserves that water in the soil for when it is really needed. This technology also helps us tackle heat stress. As global temperatures rise, plants face vapor pressure deficit. Essentially, the air becomes a vacuum sucking water out of the leaf. This system allows scientists to simulate those high-stress conditions and watch how the guard cells physically hold up. Do they collapse? Do they flutter? Understanding the failure modes of stomata is the first step to bioengineering heat-proof crops. But the implications go beyond the farm. They reach all the way to the climate models that predict our planet's future. Global climate models rely on mathematical formulas to estimate how much carbon forests and crops absorb. Currently, these models use simplified assumptions. They treat the canopy as a big leaf with average properties. They assume plants stop losing water instantly when the sun goes down. But Stomata In-Sight has shown us the reality is a messy, sluggish 30-minute lag. That means our calculations of global water cycles could be off by gigatons. By providing precise, kinetic data on how different species breathe, this technology can refine the land surface models used by the IPCC. We might discover that certain forests are less efficient carbon sinks than we hoped, or that specific agricultural regions are releasing more water vapor than predicted. This data is massive. A single experiment generates terabytes of 3D video and gas logs. To handle this, the team is using artificial intelligence. Machine learning algorithms are trained to recognize a stoma, measure its aperture, and track it over thousands of frames, removing the human bottleneck. Looking forward, we can envision a future where this moves from the lab to the field. As photonics on a chip advance, we might soon see field-deployable laser robots roaming the cornfields, clamping onto leaves to check the respiratory health of the crop in real time. We might even see synthetic biology applications, where we engineer entirely new types of stomata that are triggered by specific light wavelengths to ensure survival in extreme drought. In conclusion, Stomata In-Sight is more than just a new instrument. It is a conceptual bridge. It bridges the gap between the microscopic world of cellular biology and the macroscopic world of atmospheric science. It connects the swelling of a single cell to the hydrology of a continent. For the first time, we are not just observing the aftermath of plant respiration. We are watching the process unfold. We are seeing the hesitation, the reaction, and the resilience of the green world. As humanity faces the dual challenges of feeding a growing population and stabilizing a volatile climate, this laser-focused insight into the breath of plants may prove to be one of our most vital tools. The mouths of the plants are speaking to us, and finally, we have the technology to listen. Thanks for listening to G Fun Facts Online.